Good afternoon, everyone. My name is Dr. Obinna C of the Department of Biochemistry, University of Calabar. My topic is Cyclogossine B drug suitability against target proteins implicated in leukemia. Insight from density functional theory, pharmacokinetics, and molecular docking. Next slide, please. All right, these are the contents of the research. Next slide. The present study explored the potentials of cyclogossine B as an anti-leukemic agent. We utilize density functional theory, molecular docking, and pharmacokinetic protocols for the investigation. Next slide. For the statement of the problem, reports have shown that leukemia prevalence is on the increase in Nigeria, and this warrants, search for new drugs to help this situation. Next slide. All right, for the aim and objectives. The aim of this work is to explore the efficacy of cyclogossine B in leukemia treatment by targeting two proteins involved in leukomogenesis, that is leukemia progression. So you can see the objectives there. Because of time, I wouldn't dwell so much on that. Next slide. For materials and methods, this work is purely in silico. In other words, it is computer-based. So we utilize a lot of advanced, software application packages to carry out our research. Next slide. All right, cyclogossine B was extracted from latex of Jatropha gossipifolia. This plant is widely distributed in southeastern part of the country, and it is also found in Cross River. So HPLC, which is high performance liquid chromatography, was used to analyze the structure, and we discovered that it has 8 amino acids linked together by peptide bonds in a cyclic form. Let's look at the next slide. Yes, figure 1 there is cyclogossine B I'm talking about. The figure 2 is the prepared form for computational analysis under the framework of density functional theory. I will come to that later. Then figure 3 is asciminib hydrochloride, which is an approved drug, an approved anti-leukemic drug. So we involved this drug in this work because of comparison with our compound. All right, justification for selecting cyclogossine B in this work. The justification here is that it has structural resemblance with 2 cyclopeptides, facalistatin 10 and facalistatin 11, which have been demonstrated to have anti-cancer effect. So we looked at structure-function relationship. That was why we selected that compound. Now, next slide, please. Yeah, so we selected 2 proteins involved in leukemia progression. The first protein is dysregulated. That first protein you're seeing there is dysregulated tyrosine kinase, which is responsible for the proliferation of abnormal granulocytes or abnormal white blood cells. And of course, that is the hallmark of leukemia. The second protein there is responsible for the drug resistance experienced by leukemia patients. So we interacted our ligand with these proteins in order to destroy the amino acids and, disorganize the conformation so that we will render them inactive. Next slide. Now, we optimize our compound in 3 solvents: dimethyl sulfoxide, ethanol, and water. Of course, this is very important in medicinal chemistry because you need to know the effect of your solvent on the compound, because solvents can enhance or, mar efficacy. So the balloon-like structures you are seeing there are the electron clusters. And as drug developer, you need to know where your electrons are clustered so that you can make some modifications there. And the transition of electrons from ground state to excited state creates an energy gap that tells you whether the compound is stable or reactive. If the energy gap is high, the compound is stable. If the energy gap is small, the compound is reactive. And it is this reactivity that makes it possible for the interaction within the living system to constitute, perpetuate, and it is well. All right. In conclusion, our compound had, strong binding affinity against the, against the proteins, and this can form a good anti-leukemia drug. Thank you, and God bless all of us.